and start recording. Here we go. Wonderful. Well, welcome everyone. Thanks so much for logging in. Sorry for the technical difficulties, and looking forward to hearing Dr. Paula Bates tonight. She is just full of information around COVID and some of the research that she's done. She's also a good friend, which is great, and wanted her to share a little bit about her background and kind of show us what she's got on these slides.
Sure.
Paula?
Thank you, Tiffany, and thank you for inviting me. I did a little bit of reading and saw what the league does, and it's very exciting, very worthwhile. So I'm happy to support your efforts. Today's talk is really a mismatch of kind of stuff I do, things that I'm interested in, and that I thought I'd share with you, and maybe you'll find it interesting. I'm a big supporter of women and girls doing innovation things, and so I wanted to touch on that a little bit and just update you on some of the work that I've been doing in cancer and COVID, and in helping other people bring their innovations to patients. So I am a professor at the University of Louisville. I consider myself a longtime cancer researcher, a very recent COVID-19 researcher, and I've for a long time been passionate about innovation. And so I wear lots of hats. I still think of myself first and foremost as a researcher, even though they don't let me into the lab these days to do the experiments anymore. Mostly I'm in my office or at home coming up with the ideas and writing grants to get the awards to be able to do the research. But as well as cancer and drug discovery, a big focus of my research throughout my career has been on translating things. So taking what I discover in the lab and really trying to get it to the patients that need it. And so that's led me through this path of being an inventor, writing patents about what I've discovered, founding a company to be able to develop a discovery I made further, and a lot of times working with industry who help take it kind of the final stretch. I mentor students all the time as part of the science, but I also mentor students and faculty who are interested in doing the same kind of thing that I've done, which is take a discovery to patients or to the market. And as a result of that, I've ended up more recently directing programs that aim to help, first of all, faculty members, students, and staff at the University of Louisville who are interested in innovation, and now more recently with a program called Kinetic, a statewide program for those at Kentucky's public universities and technical colleges who wish to be involved in innovation. And so just a little bit of background, you may have picked up that I have a funny accent. That's because I was born in England in a place called Cheshire, where they speak really broadly like this. And then I went to Oxford University, where they speak in rather a posh accent, and then to London, where the accent changes again. And then in 1996, after I finished my PhD, decided to move to the United States and went over to Birmingham, Alabama to do a postdoctoral fellowship in molecular biology. My boss at the time, Don Miller, moved in 1999 to Louisville, Kentucky to head up the Brown Cancer Center here, and made me an offer I couldn't refuse. So despite the fact that I had planned to go back to England, I found myself moving to Louisville in 1999. And here I am 22 years later, 23 years later almost. And I'm consider Louisville my home. I was married shortly after moving here, and so this was my first home, and had children who were born and raised in Louisville. So I consider myself a native. And just in case you were wondering where the bit of the Australian or New Zealand twang in my accent comes from, that's from my husband, who is from New Zealand. And so a little bit about what I do. I don't want to get too much into the details of the science, but just to give you an overview of the types of thing I study. The first one is called an aptamer, and this just means it's a piece of DNA that likes to bind to specific proteins. And we found, as I'll tell you in just a second, that this can be useful for treating cancer, and now looking at it for treating COVID-19. As part of that project, kind of developing it further, we've worked with engineers to make nanoparticle versions of the same thing. And in other completely unrelated things, we work with different small molecules that we've discovered really by chance to be able to kill cancer cells without affecting normal cells. I do a lot of collaborative research with people who are immunologists and engineers and chemists and all kinds of things. And I love working with different disciplines. I think really you see a lot of innovation spark when you get two sets of people who really are both scientists or engineers, but they don't know exactly what the other one does, and you put them together and find that little place where they overlap. And as I mentioned, I've been involved in innovation programs that attempt to teach university researchers about product development. And so first question, why are people like me, cancer researchers, studying COVID-19? Well, like everybody else in the world, we want the pandemic to be over as soon as possible. I think there's light at the end of the tunnel finally. For a number of reasons, both selfish reasons. I want to get back to going out to eat and seeing my friends and hugging my friends. But for cancer patients, there are a lot more concerns. First of all, cancer patients are much more at risk of getting sick from COVID-19. And secondly, the effect of the pandemic has been that people who have cancer or people that might be at risk of cancer have not been getting their routine care that they need a lot of times due to fear of catching the virus. And so oncologists and cancer researchers in particular are keen to have the pandemic over as quickly as possible. The other reason why cancer researchers are thinking about COVID-19 is that the treatments that we use for cancer can affect the immune system. And that is obviously a critical determinant in how things work out for people who had COVID. So for example, patients who are receiving chemotherapy will have a depressed immune system. Those who might be receiving immunotherapies, their immune systems are manipulated by those therapies. But also we hope to, by understanding the way that COVID-19 interacts with the immune system, we might actually learn something interesting about cancer and its interaction with the immune system. And so that's a little bit what this is about. Talking about, in some ways, that cancer is kind of similar to COVID-19. So our immune systems can detect abnormal cells that can become cancerous. They can also detect viruses like SARS-CoV-2, the virus that causes COVID-19. And so when our immune system works properly, it protects us from things like COVID and things like cancer. But to be able to thrive in our bodies, both cancer and COVID-19, or the virus that causes it, need methods to kind of hide from the immune system and avoid being destroyed by it. And the other thing that they have in common is that sometimes the immune system, when it works well, protects us, but sometimes it can overreact or not react appropriately. And this in itself can cause problems that either allow the virus or the cancer to take hold, or even can make us sick due to the immune response. And then the second part of the overlap between these two diseases is the fact, and this affects my work, is the fact that some of the proteins that we target to try and treat cancer may actually be good things to target for treating COVID-19. And so before I tell you kind of my story about how my cancer research kind of spun over into COVID research, I wanted to mention just some background on some of the other things I mentioned. The first is the Brown Cancer Center. So I work as a researcher in the Brown Cancer Center. I'm not in the clinical building. I'm in the big clinical translational research building, which is right next to it. But if those of you who are not familiar, it's a really a fantastic place, both for care and research. One of the things that we really are leaders in is cancer immunotherapy. And that's a way to essentially take the brakes off the immune system so it can recognize cancer and start attacking the cancer. And this has really revolutionized cancer care. About 40% of patients who have cancer now are eligible for treatment with immunotherapies. And a big part of what we do is trying to understand how cancer hides from the immune system, what are the mechanisms it uses, and how can we attack those mechanisms to essentially unmask the cancer and allow the immune system to eradicate it. And we've just got a very big grant just about a year ago now, actually, to really push that forward, to harness the power of the immune system to treat cancer. And then the next part I mentioned was these innovation programs we have. And I really like to highlight what a powerhouse the University of Louisville is in the country at doing innovation. And I don't want to go into the details of all these programs, but these are very prestigious national programs that have been awarded to the University of Louisville. So some of them, there are only 3 in the country given out, and the University of Louisville was the one of the places that received one of those. And so what these awards do for the most part, one in particular, the ones that I'm involved in, are given out by the National Institutes of Health, which is the major funder of biomedical research in the country. And you've probably heard Dr. Fauci is the head of one of the institutes of the National Institutes of Health. But the NIH, as we call it, gives out about $30 billion a year in research grants to universities. And researchers like me use that money to really study how things work, what are the mechanisms that causes disease and so on. And what we produce at the end of that is literally tens of thousands, perhaps even hundreds of thousands of research papers. And you're like, that's lots of knowledge, lots of ideas, but research papers are not going to cure cancer or prevent us from getting COVID. And what we want is for those ideas written down in research papers to materialize into actual products, into new drugs, new tests, and new devices that we can use to get better. And that process is very slow and inefficient. Out of those potentially hundreds of thousands of papers we produce, maybe 1 or 2 get turned into something useful directly, and it takes a long time. And so we're trying to address that kind of barrier. And there are a number of things that make it slow and not very successful. One is that there's not money to do that kind of critical research that tests a new idea. The second is a lot of researchers in universities don't know how to do that kind of research. And so we provide those things and try to help move them along. We are, again, one of the things I'm really proud of is that Kentucky is really on the map, along with these other kind of big name institutions, as a place for doing this kind of research. And 2 of those are involving the University of Louisville. And so our statewide program now is a collaboration between the University of Louisville and the University of Kentucky, which in itself is a pretty miraculous thing. We work very well together. And we also include all of the other regional universities in Kentucky, as well as the Kentucky Technical and College community college system. And so these are grants that are available to anybody, students, staff, faculty in these universities and community and technical colleges, to try and take any idea or discovery they've made and turn it into a useful product. And this is the first program that we run in Excite, and just to kind of illustrate all the exciting things that came out of that program. Several new companies with developing products that are very diverse. Everything from, we had one group that was developing an anesthetic for eyes. People often scratch their eyes, they're very sensitive, it's very painful, but there's really not any kind of anesthetic you can put in your eyeball without kind of dissolving it. And he has found one. Another group is developing what we call the singing baby bottle. So babies often have trouble transitioning from breast to bottle feeding, and this is a device that helps them do that. This group in the middle developed a way to store blood on the shelf at room temperature. So as you know, probably blood only lasts for a few weeks and then we have to throw it away. And there's this mismatch in supply and demand that really can cause a problem, especially at times like this during this crisis. And so they've figured out a way so that you can freeze dry the blood and essentially put it on the shelf. And when you need it, you add water and shake it up and it comes back to life. So it's pretty cool stuff. And then we have an anti-inflammatory and a test for heart attacks. And so I want to just switch gears a little bit now and talk about how I got into COVID-19 research. This started actually more than a year ago. As soon as I heard at the beginning of last year about this new coronavirus, I started to think, oh, that doesn't sound good. Um, and I hatched an idea based on some of the work that I'd done in cancer involving a protein called nucleolin, which is a protein that's involved in helping cancer cells to grow, but it's also a protein that is involved in mediating the effects of various viruses. And so here's this protein. It's in most cells, it's in the center of the cell and the nucleus and the nucleolus. But when cells get stressed, it kind of comes out and moves towards the outside of the cell. And in cancer cells, they kind of, it helps them survive. So they hijack that and they have tons of this stuff, including on the outside surface. And so we had developed a treatment for cancer initially that bound to nucleolin. It's called AS1411. And we had taken, this is many years ago now, myself, Don Miller, and John Trent had developed this treatment and taken it into clinical trials for cancer, shown that it was very safe and that it had a little bit of effect, but really not enough of effect for it to kind of go further. And at that point, we carried on working at improving the pharmacology of the drug so we could make it better. In the meantime, we and others have been researching nucleolin and found out that it is involved in the replication cycle of many different viruses, including other coronaviruses. And so my idea back kind of this time last year was, can this nucleolin aptamer that we've developed called AS1411 block infection by SARS-CoV-2, which is the virus that causes COVID-19. And anywhere else most other places in the country, the idea would have stopped there. Nice idea, maybe it would work, maybe it won't. But because we're in Louisville, we have a great facility here that is headed by my colleague Kenneth Palmer. We have what's called a regional biocontainment lab, where they're able to work with viruses such as COVID-19. It's not something that I can get in my lab and just go around testing because of the safety issues. And so Kenneth was able to test this. I didn't want to show you kind of too much, science and graphs, which I thought might put you to sleep. But I just want to give you an example of the kind of things. This was what I call the eureka moment. And this was really the first experiment that they did. And so what you see on the left that looks like just a pink square is actually a dish full of cells. And if you look really closely, you can see there's, that kind of, speckled pattern is the cells. They're all growing, they're covering the plate. And those of us who look down microscopes at cells all the time will tell you that those are happy, healthy cells. In the middle, what you see is what happens when you infect the cells with the SARS-CoV-2 COVID virus. You see, excuse me, there's far less cells. They're kind of ratty looking, they're clumped together. And again, those of us used to looking at cells down the microscope will tell you that they're not happy cells. They're dead and dying cells as a result of being infected by COVID-19 virus. And then what you see on the right-hand side is what happens if you do the same thing, but this time you add our potential drug AS1411. And you can see now you get back to looking like you were at the beginning. That pink square is in fact a dish of happily growing cells. So you've blocked the virus from infecting those cells and killing those cells. And so this is, where we are right now. It's looking very promising. I didn't want to, show too much data, but we have done additional studies showing that it works first in cells and then in human tissues, lung tissues, and then in animal models of COVID-19. We know from our previous work that the drug seems to be very safe. It's been tested already in about 100 patients with cancer in the past and had no serious side effects. And so we're very hopeful that we can, move this quickly and test it in patients who have COVID. And even though we're all hopefully going to be vaccinated very soon, if not already, I think there's still going to be a need for drugs to treat COVID as we see kind of outbreaks come up over the next few months and into the future. And so right now we're working on scaling up the manufacture of the drug, completing those studies and getting FDA authorization for human studies. And at the same time, we're actually working with the same company, which is called Collagen. It's a company out of California, to go in the other direction with the cancer drug, the nanoparticle cancer drug that I mentioned for the treatment of cancer. And last but not least, I wanted to acknowledge our company here in Louisville, UPS, who provided actually the initial funding as in the form of a gift to do these AS1411 studies. So I think that is it from me. I will say I am very, very happy to answer any questions at all about what I do. I'm happy to try and do my best to answer questions about COVID-19. I'm not a medical doctor. I'm not even necessarily a COVID expert, but I am, have tried to keep up with what's going on in the world of COVID science. So I'll be happy to try and answer any questions you may have.
Paula, thanks. That was, I mean, there's so much great information on those slides. And again, I'm so sad that, we had the technical difficulties, but at least we've got this recorded for people to be able to see it. Because I think a lot of your slides, and Mary, I'm glad you were able to join us. But in the beginning slides, kind of shared a little bit of history on Paula and kind of her journey with all of this. And again, I just think it's fascinating, all of the access to everything that you're able to get.
I think, a lot of people don't realize that, Louisville is pretty good at a lot of the stuff we do. I say cancer, that I'm sure it's good at the University of Louisville, I'm sure is good at lots of things, but in my particular field, the cancer immunotherapy and this access, the availability of doing this type of COVID research is pretty unique in the area. So
yeah, and I don't know if a lot of people, I mean, they know the Brown Cancer Center is there, and they see the awards and all that, but I don't know if they really realize.
Yeah.
How
it's really, having a research center. People ask me all the time, would you go there if you had cancer? And honestly, it would kind of, it would depend, if I had maybe an early stage, not very scary cancer, I might go wherever was most convenient. But if I had, uh, something where, it was advanced, or I would really go there just because they have, all, and I'm sure there are lots of great places, but, they really are the ones doing the research, kind of pushing the forefront of the clinical treatment, especially in terms of immunotherapy. So.
Sure. Now I've gotten one shot. I don't know if anyone else has gotten theirs, and I got the Pfizer. I didn't know if you had any thoughts around, because you keep hearing people saying, oh, the second shot, affects everybody a little differently and all of that.
Yeah, and so I'm with you. I've got my first shot of Moderna, and I was feeling a little rough the next day. Honestly, I had a bit of a headache. I went to bed at 7 p.m. And I have heard that from several people who've had the second shot that it's they felt pretty crummy for a couple of days, but I think it's all worth it, right?
Oh, sure.
Just to know that we will be protected. So
exactly. Beth or Hannah or Mary, any questions? I think you guys.
Well, I just want to say that I think the James Grant Brown Cancer Center is well known in this community. I know, Tiffany, you're rather new to Louisville, and I've been here for a long time. And the Junior League has had numerous partnerships over the years with different components of the Kentucky Cancer Center and the breast cancer program. So I think for us older sustainers, of which I am, you two aren't. I don't know Hannah. I don't think I know Hannah. I think it's well known, and like you, Paula, I'm part of the Norton Healthcare entity, and or that's where my doctors are, and Norton's has a cancer center. Fortunately, I've never had to experience. I've had the experience of cancer myself, but I feel like I would probably have to go to my own doctor first, but I would go to the Brown Cancer Center just because of the knowledge and experience and research, and that says that I know more about that facility than I do what's connected with Norton's, which is probably a plus or minus. I guess my question is, and I was trying to get on via my phone, which just didn't work, but with this, one of the slides said something about FDA like approval or something. So what you've been working on, so the next step is to go through the FDA approval process, and if they approved it, then what?
Yeah, so this has been a real eye-opening experience. We've done this before for the cancer drug, and I'm doing it again right now for treatment of COVID-19. But the way it works is you have to get FDA approval to even start the human clinical tests.
Oh, right.
And so you have to show the FDA that that you file what's called an investigational new drug application, and it if it was actually on paper, I mean, it would be as tall as I am. I mean, just they ask for just this incredible amount of information, and that's why when people talk about the safety of the virus, the vaccines and so on, and the safety of drugs, the FDA are like, I mean, they are on it. They want so much information. It's not just about the kind of things I showed to show that it works, and there's loads of safety tests, but even just like the way you manufacture it, because you could do a test and show that this drug that you made is safe and it works, but can you be sure you can make it exactly the same way each time? And everything from the type of glass vial you use to the compatibility with the tubes that you put it through, just they are so picky about every single thing, which is is a really good thing. It makes me feel that anything that gets FDA approved to go forward even into clinical testing or for human use is really going to be very safe and have gone through a lot of really rigorous testing. But when you're part of the team that's trying to push it through, you're like, oh please, no more studies, we just want to get this into clinical trials. But we're very close to filing that final document needed. So.
Great. Beth, any questions from you or Hannah? I think Hannah's trying to connect.
Really fascinating. I enjoyed so much listening to the research portion, as well as all of the really neat things that are occurring to bring ideas from all of these research papers to fruition. I mean, it's just super fascinating. So I appreciate your time today, because I learned a whole bunch of new stuff, not only in how you approached bringing this therapy that was originally targeted to cancer to COVID, but also just some of the other neat ideas that are able to come to fruition due to U of L taking this step forward. It's just fascinating. So yeah,
thank you.
Very appreciative.
Great. Hannah or Mary, anything else?
No, I just like this. I got second what Beth said, the
Sure.
kind of interaction, and that gives a lot of credit to U of L and the research centers that are located with U of L, that started years ago with I can't remember who, but and there are a lot of subsets of the research that a number of friends and junior league members have worked at. The one med center or something where they had all the hospitals working together and collaboration and doctors from the various disciplines working together, which was at the time unique to Louisville. I don't know whether other cities have capitalized on it, but I have a sister-in-law who was she has a degree in microbiology, environmental health, and every once in a while she'd send me something and say, do you know this is going on in Louisville? We know about it from IU Medical School. And I'd say, yeah, because I knew somebody that was working in that field. So anyway, so at least it's known about in Indiana. So
well, we tried to get the word out about all the cool stuff that's going on. And
yeah, I love the what you showed about the different things that are up and coming for from the innovation. And what was it called again, Paula? The kind of innovation.
The program, the initial program was called Excite, which was, I can't remember, some strange acronym I came out with: expediting commercialization, innovation, translation, and entrepreneurship. But it ran for 5 years, and it was a real success. Part of what we tried to target was not just, it's like any kind of investment. You're not everything that you invest in is going to be a winner. And we try to target changing the mindset, both of the researchers who are doing it, because we're taught to think about, going very slowly and carefully and publishing papers, winning grants. And it's that whole, we don't want you to publish a paper. We want you to, develop a new medicine. And how do you get there really quickly? And it's almost like you go from trying to prove your idea is right to trying to prove yourself wrong. Because if you're going to be wrong, you want to find out as quickly as possible. And so it's really a different way of thinking. And then also just the institutional mindset too, in that a lot of the people who got these awards had never done anything like this before. They were maybe the first in their department to do it. And one example was the engineer on the blood team. And he had never done anything like this before, but now he goes back to his students who are doing engineering, and he instills in them this same way of thinking about how do you take what you discover or an idea that you have and try and translate it to help patients or help society or whatever it may be. And so we really try to, have an impact. And I do think the University of Louisville is unique in the way that it supports this type of thing. A lot of universities don't. They want their faculty to be publishing papers and getting grants, and they don't care. They don't really, give a lot of credit for forming companies or writing patents and things like that. So it's good that we have those opportunities here.
Well, I think just what you said, the integration among departments. They have the engineering department, and that's kind of the 3D machines today. First they were built to do copies, and now they are making instruments.
They can print a heart, right?
Print masks and whatever. So
yeah, it's pretty amazing, all the innovation that's happened over the years. And it's just neat for us to be able to see hands-on kind of some of the things that you've done with all of that. So just really appreciate the time. And
if you have ideas, come talk to us. So the next thing we're hoping to do is really kind of try and get out more into the community and have, encourage community innovators and community businesses to work more closely with the university. And again, I think it's anytime you get people from different, walks of life, just putting their heads together, maybe one has a great idea, but doesn't know how to do the science, or maybe the scientist has a great idea, but doesn't know about the business aspects of it. And so, I think there's a lot of can be done by bringing people together. A lot of it's amazing. Another talk I give is about where does innovation come from, and it's amazing how many life-changing innovations come from serendipity. Just people just did something. All my entire career is actually based on accidents. So the AS1411 was never actually truly designed to do what it does. It was supposed to be something completely different, and we discovered it, just like we discovered when it was a cancer drug, it treated COVID. Actually, it wasn't intended as a cancer drug even in the first place. It was something completely different, and it ended up treating cancer. So my entire career is based on these serendipitous discoveries, and so I don't know how sustainable that is, but it's worked pretty well so far.
So where do you come up with these names like the AS414? Or I've always wondered that because one of my friends is on a lot of medication and let alone she can't pronounce it, and then she wants me to call and renew it, and
Yeah, so there's a whole story behind the way they name drugs, and so a lot of these names, the numbers and letters are just the code names for that the companies give them. So this was developed by at one point by a company called Antisoma, so that's where the AS comes from, and then they just give them a random number, and they don't want to call it Antisoma 1 because it sounds like they don't know what they're doing. This is the first thing they've ever made, so then they give it some random number like 1411. And so it's kind of funny you should ask, because just this week they decided to change the name again, and so now it's because it's being developed by a company called Qualigen out in California. So now it's going to be called QN another number. So I have to get
formerly known as AS.
Formerly known as AS 1411, and before that was known as Agro 100. So.
That's interesting.
And yeah, it's hard, but it's hard when you publish because you want people to, the companies don't care. The companies don't want people to be thinking of what it was called before because now it's their drug. But when you're the scientist who's been working on it through all of these name changes, you want people to understand that, this AS 1411 is the same thing as agro. And so like Mary said, it's, QN, whatever number it is, formerly AS 1411 and formerly before that, agro 100.
Oh my gosh. That is really interesting. And I mean, it does make sense because they do want to probably have it to the latest and greatest that's helping with the funding and everything else.
They've been very good. They've been very good partners. So
fantastic.
Tiffany, when you scheduled this, did you realize it was spring break?
No. But we had some technical difficulties. Unfortunately, we had like 10 to 15 people that we're going to attend, but that's okay. We keep trying.
Well, we're the lucky 5 or how many?
I think so. Because I think
so now we can carry forth and promote it. And
the rest of them can watch the reruns, right? Just like we all live in this Netflix culture now where whatever we want to watch is completely on demand. So exactly. We can fast forward through the boring bits.
Exactly. Well, I just wanted to thank you, Paula. We're right at 7:30, which is what want to be cognizant of your time and everyone else's. And like I said, this has been so informative, and I think everyone, would agree. There's so many nuggets in those slides that you put together for us. Thank you.
And before you go, I have to ask Beth, where is the photograph behind you? Is that the Charles River?
So this is in Paris, I believe, which I've never been, but my husband went once with he played football at Hanover. So he went and then came back and bought all of these beautiful photographs, and they've just stayed with us.
It looks very European.
For, I guess we've been married 13 years and he went maybe 5 years before we got married. And this is actually not where I usually do Zoom calls. My son, my oldest son, our whole house is a classroom now because, of course, JCPS has been shut down for quite a while. So this is where my oldest son does all of his work. So it's a pretty good backdrop.
Very good. So I'm wearing U of L, of course, Paula. So
I know, you are like all decked out.
And all I want to say, it's a good thing that U of L women are playing the second game tonight, as opposed to, good job, Beth, as opposed to the first game, or I wouldn't have been there.
Well, I'm glad too, Mary.
So
it's funny.
And the UK women played great. They just didn't play as well as, if they played better than U of L played their first 2 games, but U of L just kind of had the luck of my Irish was flowing to them, but they played great on Sunday, and they play tonight at 9:00 against the Stanford Cardinals.
Dana had a game.
What?
Hopefully Dana plays Dana, their
Oh, yeah.
their little
I mean, hopefully she
feisty point
continues. I
guard
mean,
is
she
just
finally-
amazing.
She finally got her whatever you call it, her steam back or her skill back on Sunday. So anyway, well, thanks again, and we look
right.
forward to the link
Enjoy the
so
game.
I can share that with others. And
Yes, definitely. Thank you, Paula. And
Bye.
hopefully we'll be able to have you again at some point where you've got this approval for all this stuff.
Hopefully.
That would be
fingers
kind of a
crossed.
neat thing for us to do a little follow-up around that. So, but thanks again, everyone. Have a good evening, and we'll see you soon.
Thanks
Okay.
for coordinating, Tiffany.
Uh-huh.
Bye.
Bye-bye.
yeah.